Hi everyone, my name is Yasafo Amankwa and today I'll be presenting on the molecular mechanism of GRP94 in protein remodeling. So proteins are the most abundant macromolecule in the cell, performing most of the cellular functions such as signal transduction, transport of other molecules, and cellular defense. So for these proteins to perform these distinct functions, they must fold properly into their native state. In the cell, protein synthesis begins on the ribosomes as linear unfolded polypeptides, and for some proteins, they can spontaneously fold on their own into the native state. For others, they require the assistance of proteins known as molecular chaperone that will assist them to fold into their native state. Additionally, stress conditions in the cell can cause some native proteins to become misfolded, and once again, these misfolded proteins will require the assistance of molecular chaperones to fold again into their native state. In the absence of molecular chaperones, unfolded and misfolded proteins can interact with each other and form aggregated proteins, which are very dangerous to the cell as they have been implicated in diseases such as cancer and Alzheimer's. So our lab focuses on the molecular chaperone known as GRP94. GRP94 belongs to the highly conserved ATP-dependent molecular chaperone family known as the heat shock protein 90 family. In higher eukaryotes, HSP90 isoforms are present in the cytosol, the mitochondria, and in the endoplasmic reticulum where GRP94 is located. In the ER, GRP94 promotes the folding and assembly of secretory and membrane proteins and also targets misfolded proteins for degradation. In vivo studies have shown that GRP94 serves an essential function by promoting the folding of proteins such as immunoglobulins and insulin growth factors. Additionally, GRP94 plays a vital role in cancer progression by assisting cancer-related proteins in folding. Thus, GRP94 serves as a promising therapeutic target for the treatment of cancers. That is, by inhibiting GRP94, it is possible to reduce the growth or progression of cancer cells. So while less work has been done specifically on GRP94, extensive work has been conducted on other HSP90s. In terms of structure, HSP90s assemble as a homodimer with each monomer consisting of a nucleotide binding domain, a middle domain, and a C-terminal binding domain. In the apo state, GRP94 exists in an open conformation which allows it to interact with a variety of client proteins. ATP binding to the nucleotide binding domain will drive large-scale conformational changes that will allow the protein to move from the open to the closed conformation. During ATP hydrolysis, HSP90s may assume an asymmetric shape which allows sequential hydrolysis of ATP. The first hydrolysis in one arm may lead to the remodeling of client proteins, whereas the second hydrolysis in a second arm may lead to the release of client proteins. So despite the fact that HSP90s are highly conserved both in sequence and in structure, there exist significant differences between GRP94 and other HSP90s which suggest that the mechanistic action of GRP94 may differ from other HSP90s. Thus, the main objective of this project is to characterize the nucleotide-induced conformational changes of GRP94 and to see how that kind of compares to other HSP90s. And so these studies are particularly important because by capitalizing on the mechanistic differences that may exist between GRP94 and other HSP90s, it is possible to selectively target or inhibit GRP94 while leaving other HSP90s intact. So moving on to our research methods, we employ site-directed mutagenesis to introduce cysteines at desired positions, and then these cysteine mutant versions of GRP94 are introduced into bacteria. Large cultures of the bacteria are grown and protein expression is induced. The protein is then purified by affinity chromatography and size exclusion chromatography, and protein purity confirmed by SDS pH analysis. The pure protein is then made to react with spin labels that covalently attach to sulfhydryl groups of cysteine residues, and any excess free label is removed by buffer exchange method. The conformational dynamics of the protein is then detected by a method known as double electron-electron resonance EPR spectroscopy, which measures the distance distributions between two spin labels attached on a protein. So moving on to the results, We use the computational approach to select a suitable site for spin labeling. Since GRP94 assembles as a homodimer, labeling a single site will yield a double labeled protein, and so DIA measures the distance between these two spin labels. For our experiments, we selected residue 87, which has an interspin distance of 79 angstroms in the open state and 24 angstroms in the closed state. We then went ahead to purify wild-type GRP94 in the single cysteine construct, and for both proteins, we're able to purify with a protein purity of greater than 90%. Then we went ahead to confirm the successful labeling of the GRP94-87C construct as observed by a broadening of the spin label line shape when bound to a protein compared to the sharp peaks of the free label. So moving on to our DIA results, we collected DIA data in both the APO and ATP bound state. In the APO state, we did not detect a DIA signal above background. Based on our T2, which represents the lifetime of our signal, the maximum distance for which a DIA signal can be detected is approximately 60 angstroms. Our expected interspin distance in the APO state is 79 angstroms, which lies outside this allowed DIA distance. So then this result suggests that all of the GRP94 is in the open conformation in the APO state. After adding ATP, we observe a DIA signal that corresponds to a distance of approximately 30 angstroms, which is in agreement with our expected distance of approximately 25 angstroms. However, we did observe that our modulation depth, which in simple terms represents the number of spin labels talking to each other, is low. This will suggest that most of the population of GRP94 is still in the open conformation even in the presence of ATP, with only a few of the population moving into the closed state. So next, we tested the ability of GRP94 to hydrolyze ATP in comparison to the E. coli HSP90, also known as HTPG. Since dimer closure, that is movement from the open to the closed state, is the rate-limiting step for hydrolysis of ATP by HSP90s, we hypothesized that a higher ATPase activity will correlate to an increase in dimer closure and vice versa. So our results show that the ATP hydrolysis activity of GRP94 is very low as compared to that of HTPG, and so this results kind of corroborate our DIA results, which suggest that even in the presence of ATP, most of the GRP94 population still exists in the open conformation, and only a few is able to make a transition into the closed state to hydrolyze ATP. So in conclusion, our results suggest that GRP94 largely populates the open conformation in both the apo and nucleotide bound state, and this means that the ATP-induced conformational changes of GRP94 may differ from other HSP90s. In terms of future directions, we have selected new labeling sites for our DIA experiment, and we also hope to test the influence of other nucleotides such as ADP and substrates on the conformational dynamics of GRP94. Thank you.